Okay, well, welcome to part 2 of this paper, which is paving the way for adequate myelination, the contribution of galectin-3, transferrin, and iron. So this is part 2, and part 1 I started loading up earlier, but it apparently is still in the processing phase on YouTube side of things. So I posted it yesterday or this morning, but unfortunately it's still not done. Regardless, hopefully that processes before this video ends up coming out, because then there might be a little bit of confusion. So first thing that I want to mention is I realized that if people want to leave questions, biochemistry related questions, in the comments, that could definitely be a source of, what would the word be, inspiration for following videos. So if you want to leave questions, I could probably do videos answering those particular questions. That way everyone can be a little bit more on the same page, because I realize I went through my education, right? And so I can say things without realizing that my audience, even if my audience purposefully sought out this particular series of videos, they might have questions regarding certain definitions and such. So with that, just leave the questions down into the comments if you do have any during the video, and let us begin. So the bibliographical evidence shows that galectin-3 upregulation by inflammatory stimuli and its detrimental role in prion-infected brain tissue shows that there is Gal-3 upregulation by inflammation stimuli and its detrimental role in prion-infected brain tissue. So a role for galectin-3 has been proposed in the negative regulation of lipopolysaccharide-induced inflammation. So lipopolysaccharides, these are, I've gone over this previously, but these are saccharides or sugars, carbohydrates that have some lipo, or they have some, they're covalently bonded to some lipid. And this lipid carbohydrate combo is often associated with certain pathogens. And so when these pathogens release or have some of their lipopolysaccharides released, those are recognized by the immune system in order to cause inflammation. And this is beneficial for targeting those bacteria for your immune system. Now, granted, whenever we do some inflammatory experiments, sometimes we will just inject these lipopolysaccharides directly into the organism to create this hyper-inflammatory response, and then we can measure the different proteins that are being up and down regulated as a result. So one of the roles of galectin-3 has also been proposed in the negative regulation of this inflammation. So Gal-3 can be upregulated by some inflammation, but it is also proposed to downregulate inflammation in regards to lipopolysaccharides. So on the other hand, galectin-3 mediates the activation and proliferation of these microglial induced focal cerebral ischemia. So what is this? Let us look this up. So let us just look up the focal cerebral ischemia. So the focal brain ischemia occurs when a blood clot has occluded a cerebral vessel. So focal brain ischemia reduces blood flow to a specific brain region. So this would be a blood clot that happens in a particular region of the brain that kills a specific, or causes the death, causes the destruction of a certain region of the brain. So that is what that is. So let us get rid of that. So this is through the microglia, could theoretically induce this type of blood clotting. So galectin-3 mediates the activation and the proliferation of these microglia, which could theoretically cause these types of side effects. Now, granted, I want you all to be aware that with COVID-19, there seems to be an increased risk for these strokes and an increased risk for aneurysms to burst, right? So we do not, how do I phrase this? COVID-19 might theoretically be causing these, the proliferation of these microglial. Is it microglial? Wait, one second. Microglia? Yeah, so theoretically, COVID-19 might be able to upregulate or cause the proliferation of these microglial cells in a way that is pro-inflammatory and could theoretically cause things such as this focal cerebral ischemia. Sorry, I'm not in the medical field, so I mispronounce these things quite frequently. So moreover, when immunized with myelin OLG glycoprotein, so myelin oligodendroglial glycoprotein, also known as MOG, the LGAL3 or the mice that do not have the ability to produce Galectin-3, they exhibit a decrease in the experimental autoimmune encephalomyelitis. So frequently, this experimental autoimmune encephalomyelitis, this is when we will take brain tissue or nervous tissue and basically blend it up. And then we will add to that an adjuvant or something that will cause the immune system to go crazy against that material. And then we inject this into mice or whatever the model organism is. And so that makes the mouse ramp up an immune response against that nervous tissue that we just injected into them. Okay? So, and in this particular case, they're using the glycoprotein, the myelin OLG glycoprotein, or MOG. Okay, so what ends up happening is the immune system starts generating, say, antibodies, or they generate T cell. I don't know exactly what type of response they're generating, but they're generating a response against those proteins, and as a result, they get encephalomyelitis, which would be inflammation of the brain, and it's done through an autoimmune disease, so most likely through the creation of antibodies. So encephalomyelitis is inflammation of the brain and spinal cord, typically due to an acute viral infection. However, you can do it through autoimmune diseases as well. And I should also say, oftentimes the reason why viral infections can cause experimental autoimmune encephalomyelitis is because the viruses are able to mimic some of the proteins and peptide hormones that are within your body. And so since their amino acid structures sometimes will look similar to your amino acid structures, you end up generating antibodies against the virus, but then they can cross-react with your own body. And this can theoretically cause this EAE. So, however, these mice that do not produce Galectin-3, they exhibit a decrease in this experimental autoimmune encephalomyelitis. So we come in, we give these mice OLG glycoprotein injections, hoping that their immune system, or I shouldn't say hoping, but expecting their immune systems to cause an autoimmune disease to it. But if they do not have Galectin-3, then they have less experimental autoimmune severity, right? So this would indicate to me that this Galectin-3 is playing an important role in the pro-inflammatory response mediated by these microglial cells, which are an immune cell, if I'm remembering correctly. So microglial. Yeah, so they're a specialized population of macrophages, so they are explicitly part of the immune system. So due to the CPZ, and if you remember CPZ, this was the Cupro, I forgot the, so let's, the cuprazone, yeah, so cuprazone, this is a chemical that can come in and screw things up. Due to the cuprazone-mediated suppression of T cell function, the process of demyelination and remyelination in the CPZ model can be investigated without peripheral inflammation. So since we can use CPZ to cause T cell death, we can understand or look at the remyelination process without this other inflammation. So this is of fundamental importance, as the brain intrinsic effects are difficult to separate from the immune-driven ones in immune-driven demyelinating models such as EAE. So this experimental autoimmune encephalomyelitis, this is done through the immune system, right? And so this is why, and this particular model, since it's mediated through this autoimmune disease, it is very difficult to remove completely or to separate the immune effects from the myelin effects, right? So, well, for one, it's because the immune system is playing a persistent role in demyelinating or causing this in a progressive way. So this is why the Cooper zone model is very important, because we are able to remove the myelin, but then watch as the body tries to heal itself from that loss of myelin. So the CPZ-induced demyelination increases the number of resident microglial and, to a lesser extent, peripheral macrophages, which have the capacity to phagocyte myelin debris, mostly through the upregulation of the phagocytic receptor TREM2B or TREM2B. So, phagocytosis, in phagocytosis, this is when when a cell will, an immune cell explicitly, will eat or consume something else. A lot of times there's debris from these demyelinating events, so you need to have these macrophages or microglial cells. I'm trying to, yeah, macrophages will come in and basically consume that debris. And since the microglial cells are a subset of the macrophages, I would presume that they also have the ability to undergo phagocytosis. So, myelin phagocytosis is mediated by the CR3, MAP1, and the SRAI2, which are regulated by Galectin-3 dependent activation of PI3K. As a consequence, myelin phagocytosis is deficient in these Lgal3 minus mice. So this is important. So they're saying Galectin-3 plays an important role in phagocytosis. And since it plays such a large role in phagocytosis, if you have experimental autoimmune encephalomyelitis, the Galectin-3 is playing a role in the macrophages of eating myelin, right? And so this is important when you have destroyed myelin around in the brain. However, this is very detrimental if you're developing an autoimmune response against the myelin that is on the nerve tissue. And then you could theoretically cause way too much damage and phagocytosis of possibly even the nerves themselves. So that's interesting. I'm going to search this up, this phosphoinositide 3 kinase. So phosphatidyl inositol 3 kinase is a family of enzymes involved in the cellular functions of cell growth, proliferation, differentiation, motility, survival, and intracellular trafficking. So this is interesting that this is mediated in this particular mechanism. So these phosphatidyl inositols sometimes are used for anchoring proteins, if I'm remembering correctly. So yeah, this might be a very interesting protein to jump into, and I might do another video on that. So let's continue. So we have used a 6-week Cooper zone administration model in the adult Galectin-3 minus mice to evaluate the involvement in Galectin-3 in the demyelination process. And although our findings showed similar susceptibility to Cooper zone up to week 5 in both the Galectin-3 negative and the wild-type mice, and wild-type meaning the ones that are the normal mice. So the OPC, or the oligodendrocyte progenitor cells generated in the CPZ-treated Lgal3 minus mice, they showed diminished arborization. And I believe arborization would be the development of those branches. However, let me look this up so that we're not. So a fine branching structure at the end of a nerve fiber. So yeah, there's less of this arborization that is taking place. So they have this diminished arborization, which is a possible sign of poor differentiating capacity. So basically, we're getting more of these OPC cells, but they are not able to differentiate. So for instance, here we see Cooper zone. This is the CPZ treatment. This is causing more of these oligodendroglial progenitor cells being produced, However, there is less of the actual oligodendroglial cells being produced, okay? So cuprizome is lowering this amount. So, strikingly, while the wild-type mice, oh, and I should also mention that see all these extra branching, this would be the arborization that is taking place. So, good to know. So, strikingly, while the wild-type mice exhibited spontaneous remyelination at week 5, even though the demyelinating diet was still in place, LGAL3 mice exhibited continuous demyelination up to week 6, accompanied by pronounced astroglial activation. So the astroglia, or the astrocytes, these are just another one of the glial cells that are within the brain. So regarding the cell response, an increase in GAL3 immunoreactivity during CPZ-induced demyelination was detected in the microglial, but not in the astrocytes. So remarkably, while the cuprizome-treated wild-type mice displayed heightened microglial activation, as evidenced by ED1 expression, so ED1, this is just one of the proteins that the microglial are able to express. And also there's the sharp upregulation of phagocytic receptor TREM2B. The CPZ demyelinated LGAL3 mice showed an increase in the number of caspase-3 activated microglial cells. So this caspase-3, the caspases are used for, what would the word be? Basically, they are one of the apoptosis-related, well, they actually serve many functions, and I shouldn't go too far into depth, but know that caspases are able to, they're proteases that can activate other caspases, and so you end up with this all or nothing switch that takes place within these cells, and so these microglial cells are activating caspase-3, and I'll just look this up so that we have a good visual. So this is caspase-3, and so what can happen is the caspases can activate, and one of the things that the caspases can do is cause the apoptosis, or the programmed cell death, right? So that's just one of the things that, and see, this caspase-8 is what is activating caspase-3, and so then this will continue down and cause possibly cell death. I believe that there are other things that could, that these things can cause. Regardless, we shouldn't go too far into depth. So the behavioral studies revealed lower innate anxiety as early as the second CPZ week in both the wild-type and the Galectin-3 negative mice. And although only the Galectin-3 negative mice experienced a reduction in locomotor activity. So the LGAL3 mice actually had even worse outcomes than the normal mice by the second week. And they also lost the ability for their lost some spatial working memory, so the inability to memorize the layout as well. So all in all, our results indicate that Galectin-3 expression in the microglial cells appears to modulate their phenotype in response to the CPZ-induced demyelination. These results also showed that, and I should also mention these microglial cells, these are different from the oligodendrocyte cells. So these microglial cells, which are immune cells that exist in the brain, the few that are supposed to be in the brain, because remember, the brain is an what would the word be? An immunoprivileged site, so your immune system doesn't work as well in there. But these microglial cells, this Galectin-3 is necessary to cause its activation from their resting state into their activated state. And so their activated state is one of the things that is necessary to cause this differentiation from these oligodendroglial progenitor cells to the oligodendroglial cells. So let us go down again. So these results also showed that OPC generated in response to CPZ-induced demyelination in these Galectin-3 negative mice have decreased ability to differentiate, which could be due to the inhibitory effects exerted by impaired phagocytosis of myelin debris in LGAL3 microglial cells. So, if there is no LGAL3-, these microglial cells aren't able to consume the debris in the environment. So remember, when you are getting this damage, you are not just generating damage, but you're also trying to heal, right? So with the cuprizone model, you are generating this damage, and these damaged myelin is going into the environment, and that needs to be cleaned up, right? So that is going to require this pro-inflammatory Galectin-3 based stimulation of the microglial cells, which are macrophages. They will go in and clean up this myelin, which is absolutely important. Okay, but let's say you're not using the cuprizone model, which is just a chemical that you inject and leave. Let's say you're doing the experimental autoimmune encephalomyelitis model, where you are generating a immune response or an autoimmune disease against myelin, the myelin that is on the actual nerve cells that has not been damaged yet. Well, in that particular case, the damage is coming from the immune system itself. So in that particular case, you might not want the Galectin-3 to function properly, because that Galectin-3 might initiate or allow for more of this damage to happen in the first place. So what I'm trying to get at is, if the damage is coming from the immune system, you do not want to have a robust immune response. However, if the damage is coming from a chemical, and then you're left with this debris in the environment, then you're going to want the immune system to clean up that debris. And this is a tightly regulated, protected process, right? Because you want to consume the dead material, right? But you do not want to attack the living material. And Galectin-3 is one of the proteins that is tightly regulated to make sure that you are only eating or attacking the dead material and not attacking the living material. Okay, so, and you know, there's leakiness, right? So no system will be 100% perfect. Which is why when you mess with these proteins that are finely tuned to do one thing versus another, sometimes they could have seemingly paradoxical or opposite effects on the system you're working with. But if you understand the whole mechanism behind how these proteins are working, then you can have a better idea of how you can tune it some more. So rather than dealing with, oh, how do I phrase this? Rather than just throwing a wrench at something or upregulating something, you might decide, oh wait, I want to actually just tune this protein's activity either one way or the other, because going too far in either direction is going to cause damage that I do not want to see. So, continuing, moreover, this could also be explained by our previously informed findings demonstrating that conditioned media from Galectin-3 expressing but not the Galectin-3 non-expressing microglial cells successfully promote oligodendroglial differentiation. So this Galectin-3, it's not something that the oligodendroglial cells themselves are using to, well, they might also be using that as well, but that is not sufficient for them to undergo this differentiation process. However, if the microglial cells have this Galectin-3, then they are able to promote the differentiation of these oligodendroglial cells. So, previous manuscripts have demonstrated that an interruption of OPC differentiation might be the reason for remyelinating failure. So, if you interrupt this process, you cannot create these oligodendroglial cells, which are important for remyelinating the nerve cells. So therefore, the Galectin-3 negative mice could show a delay in remyelination due to failure in Galectin-3 induced oligodendroglial progenitor cell differentiation. Moreover, as Galectin-3 negative mice exhibited late remyelination in an environment which is not correctly conditioned by microglial cells, myelin generated de novo, or from scratch, is aberrant and appears loosely wrapped around axons. Interesting. So moreover, even if they are Galectin-3 negative, they exhibit remyelination, but it's late. And since it is late, the environment might no longer be correctly conditioned by the microglial cells, right? So the microglial cells are not just consuming the dead debris; they are also creating a way to get the environment ready to be remyelinated. So myelin generated from scratch is aberrant and appears loosely wrapped around axons. So maybe if you completely lose the myelin, I'll have to look how myelin is reformed, right? So maybe, yeah, because I'm not familiar with this. So if there is some damage, remember myelin is wrapped in a sheet. Maybe the damage starts from the outside and it slowly works its way in. So if you are able to remove all the destroyed material and then you rewrap around or cause the extension of the pre-existing myelin, that could be the method by which it employs. Interesting. I might have to look into that later. So microglial cells are known to release either neurotoxic or pro-recovery factors depending on their commitment to an M1 or M2 phenotype. This is what we're seeing up here. The microglial cells can be either in the M1 phase or the M2 phase, right? So that is a way that they can differentiate a little bit. So, and that will be the difference between, that'll depend, their commitments, what they're wanting to do will depend on whether or not they're in the M1 or the M2 phenotype, respectively. So, if they're in the M1 phase, they will be neurotoxic, or they will be releasing these neuron-killing chemicals. However, if they're in the M2 phase, they'll be releasing these pro-recovery factors, right? So they have been implicated in numerous inflammatory processes in the brain and have also been proposed to take part in the initial stages of multiple sclerosis. Interesting. So the activation and the proliferation of the resident microglial cells are regulated by several factors. In particular, Galectin-3 modulates microglial response to ischemic injury. And what is that? This would be when you do not have blood flow. So ischemia is a vascular disease involving interruption in the arterial blood supply to a tissue. So if you have this ischemic injury, well, that would be, say, like a stroke, right? Galectin-3 modulates the microglial response to that. And as mentioned above, it upregulates phagocytic receptor TREM2B during the CPZ or cuprizone-induced demyelination. So in conclusion, our results showed that Galectin-3 helps the myelination process by enhancing oligodendroglial differentiation and favoring oligodendroglial fate, following the neuronal stem cell differentiation. So the contrasting effects of Galectin-1 and Galectin-3 on the oligodendroglial differentiation could be explained in the context of their anti- or pro-inflammatory roles. So Galectin-1 is usually considered to be more of an anti-inflammatory protein. Galectin-3 is generally seen as more of a pro-inflammatory protein. However, I will also mention Galectin-3, when it's on the inside of the cell, tends to be anti-apoptotic, which it helps the cell decide to survive, and this is for the T cells. The T cells will have Galectin-3 inside of them, and the Galectin-3 is useful for allowing them to survive. However, if Galectin-3 is on the outside in the extracellular matrix, it can actually cause a subset of thymocytes to kill themselves. But granted, this is happening in the thyroid. I don't believe this is happening in the brain. Let's continue. So, like many cytokines and growth factors, the galectins may exert a two-side effect depending on intrinsic factors, including protein physiochemical properties, whether or not they're dimerized or oligomerized. They have stability in tissue, relative activity of the metalloproteases, matrix metalloprotease concentration, and cell redox state, which is how oxidized or how reduced the environment is, as well as extrinsic factors such as the glycosylation status of target cells. So target cells Remember, these are lectins, right? So the lectins bind to carbohydrates that are on these cells. So they are extrinsic factors such as glycosylation. If those proteins are glycosylated in a different way, then the galectins might not be, excuse me, the galectins might not be able to bind to them the same way. So it's a noisy field, right? There's way too many variables to keep track of. So we're just trying as good as we can to develop these models, and then we refine the models more and more as we get more information. So in addition, our findings demonstrate the upregulation of Galectin-3 in the microglial cells during this cuprizone-induced demyelination and its role in the regulation of microglial activation and phenotype, hence driving the onset of remyelination. So first, the Galectin-3 has its role in regulation of microglial activation and phenotype, hence driving the onset of remyelination. So these microglial cells are not just remyelinating, they seem to be also, or at least the immune system in general, is consuming the debris as well. So there are different jobs that need to be done in sequence, and this is why this is a regulated process. So briefly, our studies prove that Galectin-3 deficiency leads to, one, similar vulnerability to CPZ-induced demyelination, but failure in triggering spontaneous remyelination. And two, there's a decrease in OPC, the progenitor cell differentiation. And three, there's aberrant microglial activation during demyelination connected to a significant increase in the number of proliferating microglia with higher levels of CD11B and caspase-3 activation, but lacking ED1 expression. So, and then four, the microglial have the inability to induce TRM2 expression or produce tumor necrosis factor alpha, which are, just know that these are cellular signals that they're supposed to be secreting but aren't. And five, the early behavioral alterations, including lower levels of anxiety and decreased locomotor activity and impairment in spatial working memory. So there are a lot of effects that these Galectin-3 negative mice exhibit. So recent work has demonstrated an enhancement of oligodendroglial differentiation in vitro with M2 cell conditioned media. So the M2 cells, remember, these are the microglia that have been activated into the M2 phase. And so we take this culture media, the media where these were growing in, and we add them to the oligodendroglial progenitor cells. Then there is an enhancement in the differentiation from those progenitor cells into oligodendroglial cells. And there is an impairment in vivo, or in the living organism, following an intralesional M2 cell depletion, so indicating an essential role for the M2 cell polarization in efficient remyelination. So if we inject something that prevents these M2 cells from populating properly, then we have an impairment of remyelination. So therefore, Galectin-3 expression in microglial could favor the onset of remyelination through the induction of an M2 phenotype or a direct effect of oligodendroglial differentiation, either extra or intracellularly. So these results may thus have important implications in the development of future therapies for a variety of demyelinating diseases, such as multiple sclerosis. So here, just note that this galectin seems to be important in these microglial cells to go from M1 into M2. But these microglial cells, as I said earlier, they will have multiple roles. One, they'll have to clean up all the debris, but then after the debris is cleaned up, then they're going to have to start this remyelination process. And so that will be a fine-tuned system, which if it gets dysregulated one way or the other, you will have an impaired response of healing. So transferrin, so finally getting to where transferrin and iron is running into this. So transferrin is the main iron carrier of the human organism, and its expression is mostly restricted to the liver. However, smaller expression levels have been reported in the oligodendroglial and the choroid plexus epithelial cells. So these are just surface cells of one of the organs, or I should say lobes or parts of the brain of the central nervous system. Gosh, so let's just look it up because we might as well know exactly what we're talking about. And what I like about Wikipedia is, okay, so this is the choroid plexus. So let us vectorization's the other version. You know what? So the choroid plexus, or the is the plexus of cells that produces the cerebrospinal fluid of the central nervous system, and the CSF is produced and secreted. The cerebrospinal fluid is produced and secreted by regions of the choroid plexus in the ventricles of the brain, and the choroid plexus consists of modified ependymal cells. So just know it's a part of the brain that is producing the fluid that the brain and nervous system gets its juiciness from. So there is some expression by the oligodendroglial cells and the epithelial cells of this transferrin, and this is of the central nervous system, and by the testicular Sertoli cells. So the Sertoli cells are the nursing cells of your germ cells. So men, these Sertoli cells are important in nurturing and providing the necessary chemicals to get the germ cells to properly turn into sperm cells. So the experimental models have shown that the transferrin molecule to play a key role during development, with the transferrin knockout mice dying shortly after birth unless treated with exogenous transferrin sources. So in turn, lack of transferrin in humans results in atransferrinemia, meaning atransferrin, the lack of transferrinemia in the blood. So there's a lack of transferrin in the blood, which is a rare genetic disorder characterized by microcytic anemia. So what is microcytic anemia? Let's search this up. Characterized by small red blood cells. So the blood cells are too small. So this is why micro, meaning small, cytic cell, so small cell anemia. So the anemia that is caused by small cells. So in the microcytic anemia, the red blood cells contain less hemoglobin and are usually also hypochromic, so they're not colored, meaning that the red cells appear paler than usual, and this can be reflected by the low mean corpuscular hemoglobin concentration. So just know that that is what microcytic anemia is, and that can be caused by this atransferrinemia and accumulation of excess iron in tissues, leading to body weakness and growth delay. So the iron is, it's not as if you don't have iron, it's just that it's accumulating in the tissues and not moving to the locations where it's supposed to be. So remember, you still have ferritin, which is storing the iron, but you need to move the iron from its storage locations to important locations. And one of those locations is into the blood cells, which I believe would be in the hematopoietic stem cells that are within the bone marrow, if I'm remembering this correctly. So that's where the iron will need to go periodically or throughout time, but if you do not have enough or sufficient transferrin, you end up having a buildup in iron in tissues that do not need anymore. So in the central nervous system, transferrin has proven to be necessary for myelin biosynthesis, and the transgenic mice overexpressing transferrin have exhibited increased myelination. So if we have a lot of transferrin being expressed, then we have better myelination, which could be very important for, let's say, therapeutics in the future. So the studies concluded by our group since 1994 first found that the intrathecal injection, and what is intrathecal injection? Let's search. So an injection into the spinal canal, so it's just into the spinal canal, of transferrin in postnatal day 2 to 5 rats. This induced the expression of several myelin proteins. So if you, and yeah, this was a long time ago. So if you take transferrin, inject it into the spine of these newborn rats, they end up having the expression of several myelin proteins, so the proteins that help produce or make up myelin. So while an ICI, intrathecal injection of apotransferrin, so this is transferrin that does not have iron in it, in these postnatal day 3 rats, increased the levels of MBP and 2', 3' cyclic nucleotide 3' phosphodiesterase, or this is a CNPase, mRNA, and protein. So without affecting those of proteolipid protein. So I believe these might be, they really should have gone into more detail as far as what these are. So what is Mbp protein? So this is the maltose binding protein, and it's part of the maltose-maltodextrin system of Escherichia cholera. No, this is not what I want. I want to have the one Mbp protein, myelin. Okay, the myelin basic protein, that's what we're talking about. So this is one of the proteins that make up myelin, and it's believed to be important in the process of myelination of nerves in the nervous system, and the myelin sheath is a multi-layered membrane unique to the nervous system. So that's the myelin basic protein that is being, so if you do this intrathecal injection, what is I? Intrathecal injection? I'm sorry, I don't know if this is the, let's just say that you put in this apotransferrin in somehow. I don't know if this is explicitly the intrathecal injection, but so if you do that with the apotransferrin, which has no iron in it, you still get increased levels of the myelin basic protein. You also get increased levels of this CNPase, and the CNPase is, I wish Wikipedia, okay. So this is a myelin-associated enzyme that makes up 4% of the total CNS myelin protein, central nervous system myelin protein, and is thought to undergo significant age-associated changes. And it has the ability to catalyze the phosphodiester hydrolysis of 2', 3' cyclic nucleotides. Just know that some nucleotides, think of ATP and ADP, those are nucleotide phosphates, right? So you can have some cyclic nucleotides where the phosphorus actually ends up making a loop. They end up making a cycle somehow, and that cyclation can be used for certain types of signaling, and this can catalyze the hydrolysis or the breakage of the 2', 3' cyclic nucleotides just to a 2' nucleotide. And through cohesive understanding of its specific physiological functions are still ambiguous. So that is just what it can do, and it seems to be important for cell signaling in some way. We'll leave that up in case that it's important. So just know that this is important in the production of myelin, and protein without affecting those of proteolipid protein. So proteolipid protein would be another one of the proteins that are necessary for myelin production, right? So the myelin proteolipid protein is a major myelin protein from the central nervous system, and it plays an important role in the formation or maintenance of the multilaminar structure of myelin. So we get a few of the proteins that are getting upregulated, but we're not getting the proteolipid protein upregulated. So I wish they would have left a list of the proteins that are upregulated through normal injection of normal transferrin. So subsequently, this injection of apotransferrin was found to increase the levels of tubulin and actin mRNA. So these are a part of the cytoskeleton. So they are upregulating the production of cytoskeleton proteins, as well as various microtubule-associated proteins. So these microtubules are associated, these MAPs, the microtubule-associated proteins, associate with the tubules, the microtubules that are produced from the tubulin polymerizing together. So, although these effects occurred only when animals were injected at postnatal day 2 to 5, in turn, the transferrin receptor expression in the in vitro oligodendral progenitor cells was proven to become undetectable by Western blot. So Western blot, this is the protein, it's a way to look at the proteins that are within the cells when cells were allowed to differentiate into the oligodendroglial cells. So the TF receptor expression in these oligodendroglial progenitor cells, these were gone by the time they were already oligodendroglial cells. So these data suggest the existence of a developmental time window for apotransferrin effectiveness. So there will only be, this apotransferrin has to interact with something, right? And so they're saying this transferrin receptor is only on the oligodendral progenitor cells for a short period of time. So apotransferrin will not be able to elicit its effects if there is no receptor. And at least if we're presuming that that is the receptor that this apotransferrin is interacting with, if you do not have that receptor, by the time it's differentiated, you can no longer have this effect. So this is probably related to the loss of the TF receptor expression in the oligodendroglial cells and are consistent with studies showing that complete transferrin receptor downregulation in mature oligodendroglial and these PND20 cells make them no longer responsive to exogenous transferrin. So the in vitro studies further demonstrated that transferrin addition to the primary oligodendroglial progenitor cell cultures promote either cell migration or maturation depending on culture conditions. So this transferrin seems to be able to not only convince these progenitor cells to tax or to move to different locations, which would be very important during, say, brain damage, so they can promote either the cell migration, the movement, or they could influence the maturation, or turning those progenitor cells into the final oligodendroglial cells, which can then do the remyelination. So in line with these findings, the transferrin administration during the recovery stages of CPZ demyelination was shown to reduce the NG2 expressing cell population, NG2 being one of the proteins, and let's look this, neuronal glial antigen 2, and plays a role in stabilizing cell substratum interactions during early events of melanoma. So yeah, just know it's one of the antigens that is present on a cell, expressing cells populations, and at the expense of an increase in the number of the APC positive cells and the myelin basic protein expression, thus promoting oligodendroglial generation and maturation. So they're saying the transferrin administered during the recovery stages, this was shown to reduce the NG2 expressing cell populations at the expense of an increase in the number of APC positive cells and this myelin basic protein expression. So what are the APC positive? Let's see, APC. I wish. You know what? I don't know what APC is explicitly. Just know that this is one of the. This is probably a protein that is expressed and can be seen on the cells, and this is associated with the cells that are able to produce the myelin basic protein. So this TF can downregulate these NG2 expressing cells at the expense of an increase in the number of these APC cells. So it's basically convincing the brain make these oligodendroglial cells to create new myelin, right? So thus it's promoting oligodendroglial generation and maturation. So transferrin inhibits the progression of OPC cycle, when using an OPC culture, the progenitor cells fraction prepared according to McCarthy and DeVillis. Okay, so these people have developed their own particular fractioning and they developed their own fractioning system for getting OPC, or these progenitor cells, out of the tissue in some way. So this premature cell cycle exit subsequently leads to early cell differentiation and favors oligodendroglial maturation. So in addition, similar conclusions were reached in hypoxia, ischemia experimental model of demyelination. So hypoxia and ischemia, these can both cause demyelination if taken to an extreme, right? So in terms of the positive effects of transferrin on oligodendroglial regeneration and remyelination. So if they did this particular experiment to cause demyelination, which remember, this would not be a progressive demyelination, this would be similar to the Cuprizone model, where you have demyelination and then the demyelination stops when you stop the hypoxia or stop the ischemia. Normally, right, that should happen. You see these positive results of transferrin causing this differentiation on the oligodendroglial cells, which then allows you to remyelinate properly. So, moreover, given that the neuronal progenitor cell, I believe NPC is the neuronal progenitor cells. I think they meant to say NSC as the neuronal stem cells, but it's hard to remember. They are potential targets for endogenous oligodendroglial generation, both in vivo and in vitro. So the effects of transferrin were analyzed on rat SVZ-derived NPC cultures. Let us look at what SVZ. So these are subventricular zone cells. Okay. So the adult subventricular zone is a region situated on the outside wall of each lateral ventricle of the vertebrate brain. Okay, so just know that that's what SVZ is. And so they took these neuronal progenitor cells from the rat SVZ, and the effects of transferrin were analyzed on this cell culture with results rendering an increase in the neuronal stem cell proliferation. Okay, these data are in line with findings described by Sargent Tanguy and others, who demonstrated a higher expression of the TF receptor, the receptor for transferrin, in rat embryo NPC compared to neurons and astrocyte cultures. So these neuronal progenitor cells have higher expressions of this transferrin receptor that is absent in the differentiated neurons and the astrocytes. So this would indicate to me that these potent stem cells, or the cells that still have the ability to differentiate much further, they have higher expressions of this receptor. Therefore, they're likely using transferrin as some form of signal. So, and these people propose that the transferrin receptor is a putative or a possible neuronal stem cell surface marker. So what seems to be possible is if there are cells that are expressing the transferrin receptor, those are probably cells that have the ability to differentiate. So this might be interesting in the future if we, say, want to turn some cell types, some nervous cell tissue, if we want to make stem cells out of them. One of the ways that we might be able to accomplish this is by inducing the production of transferrin at least temporarily in them, and this temporary expression could temporarily turn them possibly into a stem cell. So that would be an interesting research field. So the multipotent capacity of the neuronal stem cell allowed us to analyze cell commitment and oligodendroglial maturation in vitro as well. The exogenous administration of transferrin to these cultures under differentiating conditions revealed an increase in the OPC proportions and a shift towards more mature oligodendroglial cells. So we take these stem cells, we culture them, and then we watch them differentiate into their different nervous tissue cell types. If we administer transferrin to these cells, it will theoretically interact with their transferrin receptors, and as a result, convince more of them to turn into the oligodendroglial cells. So Gardia, Classi, and others demonstrated similar data in vivo in a model of hypoxia ischemia, where overall myelin was restored after the transferrin intrathecal administration. So inject the transferrin into the brain, into the nervous system directly, and you see more myelination, or you see the myelin being restored after these strokes. So the intranasal administration of transferrin was found to exert similar effects by increasing the SVZ-derived oligodendroglial precursor cells. So this transferrin seems to be playing an important role in going from these stem cells to the OPC cells. So let us go here. So here we have our neuronal stem cells, and some of these neuronal stem cells are going to want to differentiate into different things, and they can differentiate into many different types of neuronal cells. But if, let's say, we want them to differentiate into these oligodendro precursor cells, which would be necessary to get it to this stage before it can go to these oligodendroglial cells, well, transferrin is playing a role in turning these stem cells into these progenitor cells. Okay. So let's go back down. The effects on transferrin on myelination are mediated by the functional convergence of signaling pathways associated with oligodendrogenesis and oligodendroglial maturation. So in terms of the downstream mediators acting after the transferrin binding to its receptor, our studies in primary oligodendroglial precursor cultures have demonstrated that oligodendroglial maturation depends on FYN kinase activation, as well as the FYN MBK ERK and the P13K AKT pathways. So a lot of bullcrap, or not bullcrap, but a lot of jargon right there. Let's first look up what the FYN kinase is. So let's search Google for this. So kinases are proteins that can take a nucleotide triphosphate of some form and take one of the phosphates off of it and add it to a protein. So these kinases, this particular kinase is also called the pro-oncogene tyrosine protein kinase, FYN. So let's see. So FYN is a member of the protein tyrosine kinase oncogene family, so they will add phosphates to the phenyl group of tyrosine. So tyrosine is one of the amino acids that make up proteins, and it has a phenyl group, and this phenyl group could replace the hydroxy portion of the phenyl group with the phosphate group. So it encodes a membrane-associated tyrosine kinase that has been implicated in the control of cell death, and the protein associates with the P8 subunit Phosphatidyl Inositol 3 kinase. So yeah, I think that was one of the, let's look up, yeah, phosphatidyl inositol 3 kinase. So this is a protein that can add phosphates to phosphoninositides. So let's see. Yeah, so they can phosphorylate the 3' position of inositol ring. So inositol, that's a sugar-like molecule, but I might have to do more another video on those as well, but we'll leave it at that for now. So just know this kinase can cause activation of these pathways, and the activation of those pathways can initiate maturation of the oligodendroglial cells. So further experiments using a dominant negative isoform of Tau to disrupt natural Tau-FYN interactions. And Tau, this is one of the proteins that make up microtubules, if I'm the tubule subunit, if I'm remembering correctly. This showed that the functional FYN is a necessary component for the OLG morphological differentiation. So changing the shape of the cell is important in its differentiation. And so the Tau proteins are necessary for the proper cytoskeleton rearrangement, right? And so the FYN has an interaction with Tau that theoretically could change its shape, right? Anyways, the oligodendroglial progenitor cells have also shown to incorporate apotransferrin through the clathrin AP2-mediated endocytosis. So just know that transferrin, normally, when it's reaching its receptors and it's trying to be, let's say if the cell needs iron, what happens is the transferrin with its iron will bind to the transferrin receptor, and then there will be endocytosis where the cell will consume that transferrin and turn it into, basically make a little bubble. And then that little bubble has the conditions to remove the iron and to basically take that iron and put it into the cytoplasm where it can be used as a nutrient, right? However, these oligodendral progenitor cells have the ability to also do endocytosis of the apotransferrin, or transferrin that has absolutely no iron in it, okay? So which indicates during iron delivery that the transferrin binds to its cognate receptors on the cell surface and enters the endosome-lysosome canonical pathway through the clathrin-coated vesicles. So as I said earlier, this is what would normally happen for iron delivery. However, the apotransferrin can also be incorporated into these oligodendroglial progenitor cells through that same mechanism. So it doesn't even need the iron to be consumed into these oligodendroglial progenitor cells. So in turn, assays performed in immortalized cell lines of the oligodendroglial lineage at various differentiation stages. So we can say, as the oligodendrocytes or oligodendroglial cells are progressing or maturing into their final form, we can actually stop it, or we have found immortalized cell lines that are stopped or halted in one stage of that progression, and since they're immortalized cell lines, they can actually multiply in that while maintaining that particular state. This has proven that the addition of apotransferrin increases the oligodendroglial morphological differentiation and maturation. The stimulation of the oligodendroglial differentiation by apotransferrin was observed to take place through the cyclic adenosine monophosphate, or CAMP, a response element CREB, or the Krebs protein pathway. And although I'll get this up, I don't want to get too far into it. Just know that the Krebs is the CAMP, or the cyclic adenosine monophosphate response element binding protein, and it's a cellular transcription factor. So what will happen is this CAMP will bind to adenosine monophosphate. Well, and remember, there's a lot of subtypes of it as well, but they are able to bind to adenosine monophosphate, and then the binding of adenosine monophosphate will determine whether or not it is active or not. So they are activated by phosphorylation by various kinases, so they can be inactive without the proper phosphorylation. And this can be done through pKa or calcium calmodulin-dependent protein kinases. So that's interesting that calcium is associated with that. So that might be the influx of calcium can also cause differentiation as well. And it's done on the serine 133 residue of this protein. And so when it's activated, the CREB protein recruits other transcriptional activators to bind to CRE promoter 5' upstream region. So this will activate certain genes to be expressed if they have the correct promoter. And the hydrophobic leucine amino acids are located along the inner edge of the alpha helix, so this is a leucine zipper. So these leucine residues tightly bind the leucine residues of another CREB protein, forming a dimer. So this is what it is. So here we have the leucine zipper between these two pieces, or these two alpha helices, and then this interacts with the DNA at a particular CREB code. If there's the CREB code in the promoter of the DNA, this will bind to it and then cause the activation or the expression of the downstream genes. So basically this will recruit other proteins to come nearby, and then it can cause activation. So the cyclic AMP response element is the response element to CRB, which contains a highly conserved nucleotide sequence, So that is what this CRE sequence is. So how is the cyclic AMP? So it gets activated by cyclic AMP. And I wish it told me how cyclic AMP is interacting with this directly or indirectly, but okay, here we go. So a typical, albeit somewhat simplified sequence of events is as follows. So a signal arises at the cell surface and activates the corresponding receptor. And this leads to the production of a secondary messenger, such as the cyclic AMP or calcium induction. Okay, so the cyclic AMP, this will be upregulated or produced in the cell, and this will serve as a signal for the cell to do a bunch of other things. So after this, which in turn activates a protein kinase, and these protein kinases translocates to the cell nucleus where it activates a CREB protein. And the activated CREB protein then binds to the CRE region. So I misspoke earlier. The CREB itself does not interact with the cyclic AMP. It gets phosphorylated, and then that phosphorylated CREB protein is then able to interact with the DNA. So, so the stimulation of the oligodendroglial differentiation by apotransferrin was observed to take place through the cyclic adenosine monophosphate response element protein pathways. So, and although CREB phosphorylation was only significant in the less mature cell lines, which this is one of the immortalized cell lines, but it was less mature, these data suggest that the cell maturational stages, at least in part, determine the signaling pathway and transcription factors through which apotransferrin induces the MBP expression, or the myelin basic protein expression. So in addition, studies in these cell lines showed that TF transferrin overexpression to promote oligodendroglial morphological complexity and maturation, and to induce the expression of several genes related to mitochondrial function and lipid metabolism necessary for myelin synthesis. So if you have the overexpression of the transferrin, it can promote the oligodendroglial morphological complexity. So remember, these oligodendroglial cells are pretty darn complex, right? And here we're saying that transferrin is the upregulation of these progenitor cells, or I should say, the addition of transferrin or the upregulation of transferrin will promote this oligodendroglial progenitor cell to turn into the oligodendroglial cell. And this is also partly dependent on what stage of maturation these oligodendroglial progenitor cells are at. Oops. So although the precise mechanism and underlying oligodendroglial maturation in response to transferrin require further description, these results contribute to elucidating the initial molecular events and transduction signals involved in oligodendrogenesis and oligodendroglial maturation. So, where does iron come in? So, iron deficiency represses one of the most Iron deficiency represents one of the most prevalent nutritional deficits, affecting almost 2 billion people worldwide. So that's about 1 in 4 people have iron deficiency. So in our studies, dietary iron deficiency during development was produced in pregnant Y-star dams, which I would presume is the name for a mother mouse, but I'm not sure. And it's by feeding them an iron-deficient diet or a control diet beginning at gestational day 5. So in contrast to the abundant evidence provided by this and other studies on iron deficiency leading to anemia, less is known about the impact of moderate iron deficiency on central nervous system development. So you could have moderate, let's say you don't have enough iron deficiency to necessarily cause anemia, but let's say how moderate iron deficiency can screw up the central nervous system development, that needed to be studied as well. So recently, Lee and others have established a model of non-anemic maternal iron deficiency during gestation, which generates slightly anemic iron deficient offspring. I would assume, I don't know how this was done, this might have been done through a mutation in iron transportation. So so that was what, 102? Go down to 1, oops. So iron deficiency disrupts axon maturation. Okay, so I don't want to go too far into all this. I would presume either they fed the mother just enough so that she wasn't anemic, but the pups still were, or there was some mutation that allowed the woman or the mother to not be anemic, but prevent some of the iron to absorb through the placenta, and that would cause anemia, slight anemia in the pups. I wouldn't know what it is, but it was it might have been down that alley. Anyways, this has established a model for the non-anemic maternal iron deficiency during gestation, which generates the slightly anemic iron deficient offspring. And these pups exhibited reduced levels of brain iron, so there was less iron in the brain, and there was significant age-specific alterations in nerve conduction, but there were minor changes in the myelin basic protein and the PLP. So this is one of the phosphatases that pyridoxal phosphatase is, I don't know if this is the one that they, let's do find PLP. Proteolipid protein. So yeah, this is a problem with having so many acronyms, is that there are, with so many different chemicals, you end up with the same acronym for the same thing. So this proteolipid protein is one of the proteins that's associated with producing myelin, right? So they had minor changes in the protein, the proteolipid protein, and the myelin basic protein, and no changes in other myelin proteins such as the myelin-associated glycoprotein and MOG. So whether acquired perinatally or during early infancy, iron deficiency has detrimental effects on brain development. So I should say these pups had very bad or significant age-specific alterations in their nerve conduction, as if the myelin wasn't able to be made properly. However, there were it looks like they're still making the myelin protein, so I'm not sure. There are only minor changes in these, but not a lot of changes. But maybe there will be more elucidation later. So, Whether acquired perinatally or during early infancy, iron deficiency has detrimental effects on brain development, including alterations in neurons and myelination, which persists regardless of and well after iron deficits have been restored. So basically, they restore the iron, and you still have these effects. So furthermore, abundant, and what I should mention is that this might theoretically be a result of, so as these stem cells differentiate into their final forms, there is a window period at which transferrin is necessary for, I should say, they lose their ability to express transferrin, right, as they mature. So there could have been a window opportunity where if you had small amounts of iron and therefore small amounts of transferrin, those small amounts of transferrin might have resulted in improper cell ratios down the road because the cells weren't getting enough of a particular signal, and so they differentiated into the wrong ratios of mature cells. So continuing, so furthermore, abundant evidence suggests that early life iron deficiency anemia may impact the development of neuronal systems implicated in neuropsychiatric disorders, in part characterized by the cognitive impairment. So, I mean, what I'm understanding here is that these creatures with iron deficient anemia, they didn't have that much different expression of their proteins that make up myelin. However, myelin wasn't produced properly, or it wasn't done correctly, and you still end up with these nervous system problems. That's what I'm understanding here. So the oligodendroglial stain for iron more robust. Oh, the oligodendroglial cells stain for iron more robustly than any other cell type in the normal central nervous system. So the iron-enriched oligodendroglial cells usually appear in rows in white matter with clearly visible cell bodies and easily detectable processes. So among others, events involving iron uptake and homeostasis are critical to oligodendroglial maturation, and their disruption can thus significantly affect myelin composition and formation. So since the foundational work by these people, the iron deficient models have targeted different aspects of deficiency, severity, duration, and developmental time windows affected. So focusing on either on prenatal or postnatal time points. So we just did a lot of research with just tweaking the different variables, and most of this was around the time shortly before or shortly after birth. So in particular, developmental iron deficiency impacts oligodendroglial maturation, which is causing long-lasting hypomyelination, which continues into adulthood even after normal iron diet reinstates. So if you do not have enough of these oligodendroglial cells, then you will end up with long-lasting hypomyelination, so you won't be able to produce enough myelination, and this is long-lasting. So even after you've added iron, even after you started producing more transferrin, that is no longer, the receptors are gone at that point. The receptors there aren't enough of them to have this significant effect from iron. Now granted, if you are injecting large amounts of transferrin into the brain, maybe that will be able to restore some of these problems, but for the most part, you've lost a lot of your differentiatability. So a growing body of evidence highlights two particular and relevant approaches in iron deficiency. On one hand, specific neuropathologies, for example, the long-term deficits in cognitive and socio-emotional function and schizophrenia, these could be modeled by iron deficiency, as they seem to involve myelin derangement. On the other hand, specific oligodendroglial requirements and the progression to a mature and myelinating state correlate with environmental nutrient availability, and thus could also be studied through ID models. So, from a cellular point of view, Studies by our group have revealed an enlarged, undifferentiated cell subpopulation in iron-deficient conditions, capable to migrate but powerless to progress beyond this precursor stage. So in other words, iron deficiency hypomyelination does not affect cell lineage specifically, but generates an arrested OPC population. So what they're saying here is that the OPCs are arrested in this location when you are iron deficient, right? So perhaps they're not actually stuck here. They might not produce enough of the transferrin receptor to then get the transferrin response to differentiate into the oligodendroglial cells. So let us continue reading. So this larger number of OPC might be a homeostatic response in order to build a complete myelin structure. The key question being why iron deficiency local OPC remained arrested. At first glance, the iron deficient oligodendroprogenitor cells appear to exhibit intrinsic alterations in terms of proliferation, migration, and maturation. So in recent years, extensive research has exposed abundant astrocyte microglial interactions with the oligodendroglial cells, which may support, limit, preclude, or drive oligodendroglial along their lineage. So in line with our results obtained from the CPC demyelination, a positive crosstalk among the astrocyte processes, axons, and the oligodendroglial cells has been reported in mixed mouse spinal cord cultures. So while dynamic changes in cell morphology and prevalence for the astrocyte differentiation around the injection site have been demonstrated in green fluorescent protein tag neurospheres transplanted into the spinal cord of dysmyelinated Shriver mice. Okay, that is a run-on sentence. I'll break that down. So a positive crosstalk among the astrocyte processes, the axons, and the oligodendroglial cells. This crosstalk has been reported in mixed mouse spinal cell cultures. Okay, while the dynamic changes in cell morphology and prevalence for the astrocyte differentiation around the injection site, this has been demonstrated in this GPF tag neurospheres, which have been, these are neurospheres that have some multiplicity in them, so they are able to differentiate, and then they have been tagged with this green fluorescent protein, so you can see the final cells that these get differentiated into after they have been transplanted into these dysmyelinated Shriver mice in the spinal cord. So they find ways to remove the myelin in the spinal cord of these mice, and then they transplant these neurospheres with this fluorescent protein and see how they differentiate in the spinal cord. So there are, at this injection site, there are dynamic changes in the cell morphology and prevalence for the astrocyte differentiation around these injection sites, right? So, and apparently there's a positive crosstalk among these different cell populations. So adding up to the description of transcription factors and molecules involved in oligodendroglial maturation, our results demonstrated that iron-deficient astrocytes exhibit an extended proliferative status. There is high basal expression of immature markers and diminished response capacity. So in addition, iron-deficient microglial cells display an anomalous cytokine profile, not only basally, but also under stimulus. So basally being without stimulus, but with stimulus, there is also anomalous cytokine profiles that they express. So, on the hypothesis that deficiencies in oligodendroglial maturation underlie iron deficiency, our group tested the effects of apotransferrin on oligodendroglial differentiation in these iron-deficient animals. So give the animals iron deficiency. Let's see if apotransferrin is able to cause something. And the sharp reduction in myelin components observed in the iron deficient conditions at the postnatal day 24, so 24-day-old mice, which this is a sign of hypomyelination. This was prevented. So this hypomyelination, which was the result of the reduction in myelin components by day 24, this hypomyelination was reduced or largely prevented by this injection of apotransferrin at day 3. So you get these iron deficient mice and you give them apotransferrin day 3 after they are born, you actually stop the hypomyelination events later in life. So when further exploring the factors involved in these ameliorating effects, cultures of oligodendroglial cells, which are isolated from these iron-deficient rats, they exhibited fewer differentiated cells relative to control. So we take the cultures of these, and there were fewer differentiated cells, as evidenced by the smaller number of differentiated myelin basic protein positive cells compared to the undifferentiated PSA and NCAM positive cells. So you had less of these differentiated cells. So additional to in vitro studies proved apotransferrin administration to have compensatory effects on iron deficient oligodendroglial cultures. So we take the cultures of these iron deficient, we take the iron deficient oligodendroglial cultures, and there is an increase in the number of the myelin basic protein positive cells and a decrease in the number of these PSA NCAM positive cells. So these are the undifferentiated ones. So if you add this apotransferrin to these cell cultures, you end up with more myelin producing cells. So this, and thus it supports in vivo findings regarding apotransferrin effects on oligodendroglial differentiation. So in this context, the iron deficient model becomes a powerful tool to study oligodendroglial progression toward a differentiated stage, taking into account not only OPC features and needs, but also their cellular environment, and thus opening doors to new targets in neuropathological approaches. So this is excellent. So this means that we could theoretically use these iron deficient models as a model for hypomyelinating events. So yeah, this is great. So and we might be able to find theoretically pharmaceuticals or nutraceuticals or different types of factors that might be able to reverse this problem even later in life. So let's say someone did undergo anemia for much of their developmental period. If these mice can serve as a proper model of those people, we might be able to develop pharmaceuticals and treatment protocols that can undo this hypomyelinating state in the mice and hopefully be able to transfer that into human sufferers as well. So altogether, our results indicated that apotransferrin has an ameliorating effect on demyelination and hypomyelination, and while Gal3 has proven to foster remyelination following the CPZ or LPC-induced demyelination. These findings encourage us to search for novel therapeutic strategies for the treatment of demyelinating diseases. So what I want to mention is that iron is playing a very important role, because without iron, you are not producing enough transferrin. So you're not producing enough transferrin, nor are you producing enough apotransferrin, so, which is transferrin that doesn't have iron. So the reason why this happens is that the mRNA of cells, there is a binding protein that will bind to the mRNA of certain proteins, right? So this binding protein, which will bind to the iron response elements. So after it binds to the iron response elements, it can either downregulate or upregulate its expression in some way. For transferrin, if you do not have iron, then this protein will bind to the mRNA and prevent the translation of transferrin. But if you do have iron, the iron will bind to the protein and kick the protein off of the mRNA of transferrin and then allow the ribosome to produce the transferrin protein. Okay? So this is why this is important. If you are iron deficient, you are not producing enough transferrin, and if you do not produce transferrin during these developmental stages, when you have a lot more of these stem cells in your brain, then the stem cell population might not properly differentiate into these oligodendroglial cells. And as a result, you are in a hypomyelinating state, or you do not have the proper morphology. To survive. So, or to have these differentiated stem cells. So, with that, I'm going to head on out, and thanks for listening. So, have a good one.